Hey everyone, welcome to lesson 7 of microbiology. So, for this lesson, we're going to be diving into what are core concepts that will really help you throughout the rest of this course. And this lesson is focused on organic molecules and microbial biochemistry. So, this is the introduction to all the building blocks that you're going to use throughout the remaining lessons. Especially in lesson 8, we're going to get into energy processing of the cells. And in that, we're going to use a lot of these different core elements that we're discussing in lesson 7. So they're kind of paired up very neatly, lesson 7 and lesson 8, a little bit more so than the other labs. Of course, they all feed into each other, but you really need to understand everything from lesson 7 to get a good solid grip on everything in lesson 8 as well. So we're going to start out with some pretty much general vocabulary and the makeup of some of our organic molecules. So if it's an organic molecule, that simply means that it's carbon based. So if you go to, the grocery store, it'll say organic, and that means it hasn't been bioengineered. If you look at organic waste, that's anything generally that is carbon based. And when we're talking about that carbon based side. Only things that are carbon based are considered organic in organic chemistry. And that also goes into you can have carbon based molecules that are, of course, carbon based, therefore organic, but not life. And those are two things that you're going to kind of have to pull apart that are conceptions that are put together for most of the time. So in this case, we're talking about carbon-based molecules, and that means organic. In our specific case, because it's microbiology, all of this is happening inside a living system that we might refer to as an organic system at a certain point, but that just means it's carbon-based at its core. Now, these carbon-based molecules are mostly created with covalent bonds, which are very stable. There's a couple other types of bonds, but covalent bonds are going to be the main ones, and they're built from monomers forming into polymers through what we call dehydration synthesis. So you're going to remove H2O to create a link, and you're going to add H2O to split. So dehydration synthesis, you're removing an H2O and you're putting something together. Hydrolysis, you're adding an H2O and that's splitting an organic molecule apart. And this is something that we're going to talk about in lesson 8, that kind of like adding, splitting scenario with a lot of the organic molecules that power the cells. Now you have some core elements, and that's carbon, hydrogen, and oxygen. So C, H, and O, those are your three core molecules. You're also going to have a couple others thrown in during certain different reactions, but when we're talking about organic carbon-based molecules, it's usually C, H, and O is what we're going to be working with. And these create four major classes of overall molecules inside the cells. Those are going to be your carbohydrates, your lipids, your proteins, and nucleic acids. So remember, amino acids make up proteins. Nucleic acids make up DNA and RNA. So those are two different things. So we kind of lump proteins together, whether it's amino acids or the entire protein, but nucleic acids we keep apart because those are the building blocks. And we don't want to say, RNA or DNA, and we just want to leave those as nucleic acids. So those are the four main things that you're going to make up. They're usually made up of a small number of functional groups. These are small parts, typically at the ends of your organic molecules, that are going to have a huge effect and determine what that organic molecule really does. You have hydroxyls, which increase polarity. So this leads to hydrophobic behavior. The reason this happens is it's a OH with a negative charge. So if you think about an O and an H together, what does it want? It wants another H so that it can become water. And this hydroxyl is the basis of that sort of lysis dehydration synthesis effect. So you can have a water come in and then, or a hydrogen rather come in and they'll create a water and then you can form two bonds between whatever's left over. You also have carboxyl ends, often referred to as the carboxy terminus of proteins. These are very highly reactive. Uh, COOHs. So if you look at that, you have a C and an O, which are highly stable together. And then you have an O and a H. Again, you have that hydroxyl. So really carboxyl end is just the OH with a CO in front of it. And then that again is highly reactive. Now there's other ones of these, but those are the two most common because they're used in that creation of organic molecules. And this is what gets us to monosaccharides and polysaccharides. So if you think about a monosaccharide, that would be your glucose, your fructose. It's immediately available energy for your cells, and it's based on this CH2O model. So you have a certain number of C's, a certain number of H's, and a certain number of O's all in one molecule. And it is the C's are multiplied by 1, the H's are multiplied by 2, and the O's are multiplied by 3. An example of this is C6H12O6. Or you could have C3H6O3. So the C and the O's are always going to be equal. The H's are always going to be double. So that's how you get your monosaccharides. So that is pretty straightforward. But then we have disaccharides. That's going to be maltose, sucrose, lactose. And that's two sugars linked by a glycosidic bond. Now, I'm not going to get into that too much. Just know that's two of those monosaccharides linked into a disaccharide. So it's just two chains linked together. Then, of course, it gets more complex, like biology always does. So you get polysaccharides. These are going to be what we can refer to as branched energy storage. So something like a starch, cellulose, uh, peptidoglycans. And those are going to be very complex and harder to break down, harder to get to that core glucose, fructose monosaccharide for the cell. So those are also very strong because they're, not having nearly as many of those reactive groups on them. So they're going to make up things like cellulose, the structure of all plant life, the peptidoglycans, those bacterial wall sugar peptide networks, and then starches, for example. Now, the roles of all of these are energy supply and storage. You want sugar because it's an easy accessed energy source. And actually, glucose, fructose, these monosaccharides, they're what your body is normally used to using and going to be using the most for energy generation, which we'll talk about more in lesson 8. Now, they're also used for some other things, which gets a little more confusing and a little more interesting. So all proteins actually have sugars on them, some sort of saccharide, disaccharide, polysaccharide on the surface, and that's used for signaling. These are called glycoproteins, and if it's a lipid, a glycolipid, and these are connected on top of the proteins and is what leads to recognition of self-protein, or at least same organism protein. And this is a way that we can tell, hey, that bacteria is definitely not from me. It has different glycoproteins. It has different sugars on it. So I need to attack that. I need to kill that and get rid of it. So it's in the wide spectrum of ways we defend ourselves from pathogens. These sugars are actually another indicator, another signal for our body to know what to attack and what not to attack. I mentioned glycolipids. So lipids have a lot of unique characteristics with very diverse structures. So we're more and more getting into that realm of I could talk at nauseum about all of these different use cases, right, of different lipids. But it is not important to know every single lipid, the same way it's not important to know every single microorganism that exists. It's just impossible. You can't carry all of that in your brain. So it's important to understand instead the base facts and building blocks so that you can take those and make very well educated determinations from a question. So if someone asked, do sugars matter for pathogen recognition? The answer is yes, they do matter because I know about glycans and all that. And then someone says, well, is fructose on a human protein or would that only be on plant proteins? Right? And that's a more detailed question that I would have to, even with all of my background and education, go double check. So you're never going to be asked details like that. It's more important to understand the basic concepts, and that'll be true more and more. So lipids are fats, phospholipids, sterols, and waxy lipids. So again, there's a lot, and we're most used to thinking about lipids as fats. So these are going to be saturated or unsaturated fats. Saturated and unsaturated, there's different terms that different people use. The general rule of thumb is the saturateds have no double carbon bonds, so they have straighter chains and they're tightly packed. The way we think about this on the day to day is you put butter out on the counter and well, in Phoenix heat, it might melt, but in a normal environment, it shouldn't melt. It should be solid. And that's because it's full of saturated fats, those tightly packed fats. Whereas unsaturated have one or more of these CC links. And when you have those links, it creates a little kink. It creates a bend, which means if you have a bunch of things stacked up against each other, and they're saturated, they're very close together. But you introduce a bend, and then it introduces spaces. So once you get those spaces, it ends up being more fluid, and then you get things like olive oil, which is fluid at room temperature, and that's an unsaturated fat. They probably have saturated fats in olive oil, it's just not enough, right, to have that solid packing. Now, there's also phospholipids, which are extremely important for your cell membranes. So they have this hydrophilic, so water-loving head, and this hydrophobic or water-hating tail. So you get these packed together, and the heads go outside, and the tails go inside, and then you'll either have one set or you'll have two sets. So you'll have this hydrophilic, water-loving heads on the outside of the cell and the inside of the cell, and then you'll have this hydrophobic space. And that's a great barrier for keeping things from going in your cells and out of your cells that you don't want. It's this wonderful barrier of phospholipids that control the flow in and out of cells. Now, sterols, they are these rigid rings of fat that they adjust that membrane fluidity. So you're going to have everything out and then everything in because of the hydrophilic heads with the water facing side and the hydrophobics in the middle that are meeting each other. But you have these sterols, and when they get in there, they actually disrupt that. So that lets things flow in and out of cells. Um, and waxy lipids, I'm not really going to touch on that is more niche even than sterols, and I think that's enough lipids for you guys. But the main things are you're going to have those saturated and unsaturated fats, and then mainly phospholipids, and that interaction with sterols is really important to understand and how fluid that cell membrane would not be without other lipids in there disrupting what's going on. Now, the major functions of lipids in cells we've already kind of touched on, but I just want to review them real quick here, is your membrane structure. That's, of course, your cell membrane, that selective barrier, and how that selectivity works. Energy storage. These lipids have so much carbon density in them that they're wonderful for storing energy long term. They're also very stable. And that's why, if you have any fat. Everyone has fat a little bit, at least. If you have any fat on your body, that's why it exists, because it's the best way we can store energy long term. It's like putting uh, milk in the fridge versus leaving it out on the counter. If you leave it out on the counter, it's going to go bad, it's going to disappear. But if you put it in the fridge, aka us storing energy in our fat cells, then it's going to last a lot longer time and be available whenever we want it, right? The other thing that fats do, again, everything is starting to do more and more and more things, and it's becoming more and more diverse, is cell signaling. This is hormones. So hormones are generally fat-based. They're going to be some sort of, well, I shouldn't say fat-based, excuse me, lipid-based, and that is a key difference. So lipids can be fats, but not all, well, I guess all fats are also lipids, but lipids can do other things than just be fat. So lipids is the catch-all umbrella, and they are also going to act as hormones for cell signaling, mood regulation, all these other things. And that is a very, very large pool to jump into that I don't really think we'll cover too much. You just need to know that lipids can be responsible for cell signaling. And again, coming back to those waxy lipids, I didn't really mention much. You have this protection from lipids because they will sort of coat things. You know, if you've ever put lotion on your hands, you get that coating, and that coating protects you from whatever it is that's in your environment that you're touching. I mean, I'm sure you've washed your hands with lotion on at least once in your life, and it's like, oh man, I need so much soap to get rid of all of this lotion because it's a protective layer of lipids, of those waxy lipids is what is usually in cells. So they're going to act in the same way. They're going to coat things and protect them from the environment. Then we go to amino acids and proteins. So remember, proteins are made up of amino acids, but usually we just refer to them as proteins as the catch-all. So your amino acids are made up of a alpha carbon plus an amino. So that is the main base, and then you add a carboxyl, a hydrogen, and the varyl group, the R. When it comes to amino acids, the varyl group, that R group, is what matters. That's what really changes things. Um, so that group think of as more of a functional group. The rest is the basis of the amino acid, and then there's that R group that changes from amino acid to amino acid. And these form into what we call peptides through a peptide bond through specifically that dehydration synthesis I was talking about. So you're going to take a water off. You're going to get connection of amino acids together, and they're going to form long strings of peptides. When peptides fold together, they become proteins. Technically, a peptide chain is also a protein, but usually we think about them in that three-dimensional space. Now, for these R groups, they can be nonpolar, they can be polar, they can be charged, they can contain sulfur, they can be aromatics. They are what's going to ultimately dictate how the proteins fold together and form their 3D structure. So they're incredibly important, but I do not expect you to learn all the R groups. Just know that there are different ones, and when you're presented with that table, do your best to clump them into their different categories. Like, is it aromatic? Is it charged? Is it polar? Is it non-polar? But this is something that memorization can be done, but it's a lot to memorize, especially amongst everything else. When we're talking about amino acids transferring to proteins, I mentioned that when you get that peptide chain, that is technically already a protein. So that's your first level of protein structure, is that linear chain is called the primary structure. Then if you have a chain and you have another chain and they come together like this, they make a sandwich, or they wrap around each other, that's going to be your beta sheets and your alpha helices. Those are the secondary structure, is these spinning helices or these flat sheets. Those then go on to fold more into your 3D tertiary structures. So that's what we usually think about when we think about the three-dimensional proteins, is they're in this complex, folded up, gunked up 3D structure. But there's also a fourth level, a quaternary structure. So primary, secondary, tertiary, quaternary structure that is multiple protein subunits coming together to form a bigger overall protein complex. Technically, those are all made up of individual proteins, but they come together in a complex 3D fold, And that is what makes your quaternary structure. So there's a lot of different proteins that do this. The one that's talked about the most is your ribosomes. So those are partially responsible for translating all of your DNA into proteins, and they come together in a large complex of many proteins, but there's a ribosome specific large and small subunit that kind of click together around your DNA and then run along it. And that's technically a quaternary structure. Now, proteins, because they're so complex, can be very sensitive and easily disrupted. So heat, pH, salts, solvents, so like surfactants, soaps, can all disrupt and denature proteins. And that's why if you've ever been using bleach and you got it on your clothes, it stains it white because all of the pigments in that clothing are protein structures. They're very usually hard to disrupt protein structures, which is why they work as pigments on clothes and you can wash them a ton, but they are ripped apart by bleach. So it's denaturing those and it's leaving that white stain where there maybe used to be a very deep, rich, vibrant color. Now, how does this really help us in microbiology, right? How does this help us identify a microbe or treat a patient or anything like that? Well, that is hard to answer with this lesson. That's more where lesson 8 comes in when we start talking about metabolites. So we talk about them a little bit here. Metabolites are anything made by a microbe as it's processing things in to get energy to replicate. Everything that comes out the other end is a metabolite. So you get things like fermentation makes acids and gases. You can detect certain enzyme activity, whether you have things that are intracellular or extracellular, where these metabolites are being pumped out, whether it's inside the cell and it's kept there or it's outside the cell and being pumped out. That can tell you a lot about what type of microbe it is, what their amino acids are that make them up. So we actually don't have the capability to make every amino acid that we need as people. We can't synthesize all of them as human beings. We need to consume amino acids, so consume proteins, and get those rarer amino acids, the ones that we can't make to actually be functional. So we require that. And the same goes for microbes. They're a lot less complex. So they might require certain amino acids to be taken in, and then they never release them. They're always using them. So even if you give them abundance, they might not be emitting those. They're always holding on to them. So that's another way you can detect and determine which microbes are which. Now, this goes into metabolomics as a subject, and that's something that's way too complicated to touch on here. If you want to get a scope of how complex the metabolism is of a microbe, go to Google, type in I, I'm trying to think. Even E. coli will look really complex. So just type in the metabolic map for E. coli and look at that, or pick your favorite microorganism, whatever it is. They might or might not have a metabolic map because they're very complex and hard to make. But think about something that you want to search up, or just type in metabolic map of E. coli, look at it for a minute, realize and internalize how complex that is and how hard it is to determine what something is just from culturing it and looking at it, and how much of a boon it is that they have these complex metabolisms that give us these metabolites that we can just look at, we can pick out, and we can say, oh, I know it's this classification of microbe, or I know specifically within this classification it's this microbe which is very, very bad, so we need to start the patient on these antibiotics. So there's some really cool stuff there with the metabolome of all these microbes, and we'll start getting into it a little bit in lesson 8, But if it really interests you, I would suggest going out and hunting some information on yourself because it's just so big that it would be very hard to cover in one lesson of this class. It's a class all its own. It is extremely complicated but extremely interesting. And I hope that this lesson has piqued your interest and you're excited to dive a little bit more in depth in lesson 8. Have a great week.